Hello, myself Dr. Humaira Tabkanatruhi. Today we will be discussing about the neuroendocrinology in relationship to the reproduction. So let's start. Here we are discussing about the female reproduction, and you know the basics about hormones. The hormones are certain factors which are released into the stream and then they are delivered to the cells. So these are controlled or secreted by the endocrine glands. They aid in the cell communication, they help to regulate the cell functions, and they also affect the other endocrine glands or the body systems. Okay, and they work along with the nervous system, hence the name neuroendocrinology is mentioned, and they promote or inhibit nerve impulses. So here the endocrine system work together with the nervous system, hence the name neuroendocrinology is mentioned, because these two work together. Coming to the feedback mechanism. So here if we study about different hormones, nervous control and all these things, you must know the feedback mechanism. Like there are two types of feedback mechanism. One is the positive feedback and second is the negative feedback. So what do you mean by feedback? Because these hormones have the feedback effect on the release of other hormones, other factors. So you must know the feedback mechanism. Or what do you mean by negative feedback, what do you mean by positive feedback? So the negative feedback, here an increase in the concentration of a hormone triggers activities that inhibit further secretion. So suppose here one hormone is increased, so its increase, what happens because of it increase, it triggers activities that inhibit the further secretion. So it whenever there is increase in the amount, then automatically there will be suppression, and this is called as the negative feedback, because this increase in the concentration is exerting a negative impact on the release of further release of the hormones. This is called as the negative feedback. Okay, so next is the positive feedback. Here an increase in the concentration of a hormone triggers activities that stimulates the further secretion. Okay, here what happens, certain hormones are increased. Okay, so here because these hormones are increased, they exert a positive effect on the secretion, further stimulation of certain hormones or certain factors. This is called as positive feedback. So negative feedback is increase in the concentration reduces the further secretion, is called as negative feedback, and increase in the concentration further aggravates the or stimulates the secretion. This is called as the positive feedback. So here you'll come to know, I'll use these words negative feedback and positive feedback while explaining the different hormones. Okay, but you must know all these things. Coming to the introduction, the neuroendocrine mechanism are the basic factors in the reproductive cycle. As I have already mentioned, these nervous system and endocrine system works together in maintaining the reproductive cycle. A transducer concept has been evolved in which the specialized neural cells of the hypothalamus function as the final common pathway to guide the appropriate anterior pituitary hormonal response. So here what happens, there will be specialized neural cells present in the hypothalamus which acts as a pathway to guide the appropriate anterior pituitary hormones. That means here hypothalamus secretes certain factors, inhibiting or releasing factors, which has a direct impact on the stimulation of the pituitary. Here the anterior pituitary is mainly involved. So here they have the effect on the pituitary. So here this acts as a transducer. So these endocrine, how they are working in together is these nerves or the neural cells present in the hypothalamus, they are having the effect on the pituitary gland. So they control the release secretions of pituitary glands. Okay, so hence this neuroendocrine mechanism is functioning to maintain the normal reproductive cycle. Endocrine system, here we will study about the HPO axis. HPO axis is, in the female it is HPO axis, in general it is a HPG axis. hypothalamo pituitary gonadal axis, whereas in the female it is called as the hypothalamo pituitary ovarian axis. So it starts from the hypothalamus. You know where the hypothalamus is situated in the brain? It's a very small in size, but it controls most of the things. It has the connections with the other parts of the brain as well as with the pituitary glands. Hypothalamus plays an important role in the neuroendocrine regulations. It has the three well-defined areas present in the hypothalamus. Those are supraoptic area. First one is supraoptic area. Second, in the middle part is tuberal area, and the third part is mammillary region or mammillary area. And these regions are further divided into areas or nuclei. Mainly the nuclei is there. Both a tonic and a cyclic center are located within the hypothalamus, resulting in the changes in GnRH production. So what do you mean by the tonic center and the cyclic center? We are going to discuss this in the further slides. Okay, so just remember this. It has a three well-defined areas and which are again divided into the nuclei. In this diagram you can see the different areas of the hypothalamus. You can see here, this is the preoptic area. This is denoted by the violet color, and the green is the supraoptic region. You can see here it's further divided into different nucleuses, and you can see here next is the tuberal region or the tuberal area. You can again see it is divided into the different nuclei, and here the next is the mammillary region. You can see with the red regions which are the regions which are marked with the red color is nothing but the mammillary region. So in this way, these hypothalamus is divided into the different regions or the different areas, and which are again divided into the nuclei. As you can see in the diagram, the supraoptic region is divided into the paraventricular nuclei, anterior hypothalamic nuclei, suprachiasmatic nuclei, and supraoptic nuclei. Whereas the next tuberal area is again divided into the dorsomedial nucleus, ventromedial nucleus, and the arcuate nucleus. You can see here the arcuate nucleus is important, and the mammillary region is further divided into the posterior hypothalamic nucleus and the mammillary body. Whereas the preoptic region is again divided into the lateral preoptic nucleus and the medial preoptic nucleus. So in this way, the different regions of the hypothalamus are divided into the different nuclei. Coming to the connections of hypothalamus, it has two types of connections. One is the cortical connection, another are the pituitary connections. So coming to first the cortical connections. The cortical connections are between the higher cortical centers and the hypothalamus. Means it has different connections with the higher cortical centers. Means with the different centers of the brain, it has the connections. Like as you can see in the diagram. So it has connections with the pituitary gland as well as with the other centers of the brain like amygdala, hippocampus, and the thalamus and the pons. You can see in the diagram it has the connections with the other parts of the CNS or the brain. That's why because of it has the connections with neural connections with the higher cortical centers, it is influencing the ovaries by certain factors like the emotional factors and the environmental factors. This is the reason we can say that the emotional factors or the environmental factors affect the menstrual cycle or reproductive system in the females through these connections of the hypothalamus with the other centers of the brain. Next, coming to the pituitary. Hypothalamus is connected with the anterior lobe of the pituitary through a special hypothalamo-pituitary portal system of vessels, whereas it is directly connected with the posterior lobe of the pituitary neurohypophysis by the supraoptic and the paraventricular nuclei. Okay, so you can see here the hypothalamus, it has the pituitary, it has two parts, anterior lobe of the pituitary, and here is the posterior lobe of the pituitary. You can clearly see in the diagram anterior lobe and the posterior lobe. So here through the anterior lobe, the hypothalamus is connected through the hypothalamic portal system of vessels. You can see here directly the vessels are coming from the hypothalamus into the anterior lobe of the pituitary. So through these portal system of vessels, it is connected to the hypothalamus, whereas the pituitary is directly connected to the posterior part of the posterior lobe of the pituitary is directly connected to the hypothalamus by the two nuclei, supraoptic and the paraventricular nucleus. You can see supraoptic nucleus and paraventricular nucleus. Through these, they are directly connected with the posterior lobe of the pituitary. So in this way, the hypothalamus is connected to the other parts of the brain and to the pituitary. In the diagram also you can clearly see how the anterior lobe of the pituitary is connected with the hypothalamus to the different portal system of vessels, whereas the posterior pituitary is directly connected through the neural connections to the hypothalamus. We can see here the pituitary. Adenohypophysis is to the anterior pituitary, including the parts like anterior lobe of the pituitary. This is denoted with the dark pinkish color, and the pars tuberalis. This is a part which is connecting the hypothalamus. Stalk-like structure is there to the pituitary, and the posterior pituitary is also called as a neurohypophysis. It consists of the stalk plus hypophyseal tract and the posterior lobe of the pituitary. You can see clearly in this diagram. Coming to the secretions of hypothalamus produces a series of specific releasing and inhibiting hormones. So here it is producing different hormones which are releasing or inhibiting in nature, which have got effect on the production of specific pituitary hormones. So these releases the specific releasing and inhibiting hormones, which is directly acting on the production of pituitary hormones. These hormones are synthesized by the hypothalamic neurons and are released at the nerve endings around the tuber cinereum. It is a part connecting the pituitary. So these hormones are secreted by the hypothalamic neurons and are released at the nerve ending. So what happens through the nerve ending, it reaches to the pituitary gland. Thereafter, these are drained to the pituitary through the hypothalamo-pituitary portal system of vessels. So first of all, what happens? These hormones are synthesized by the hypothalamic neurons, and then they are released at the nerve endings. Then what happens? When they are released at the nerve ending, they are drained to the pituitary through the hypothalamo-pituitary portal system of vessels. As I have already shown you in the diagram, you can see here how the portal system of vessels are there. So these neurons ending, they are released. You can see here the neuroendocrine cell nuclei. You can see here in the diagram, and you can also see here the supraoptic nuclei and paraventricular nuclei are directly connecting to the posterior lobe of the pituitary, and the portal systems in the anterior lobe of the pituitary. So in the diagram you can see first is the releasing and inhibiting hormones are released from the hypothalamic neurons into the hypothalamo pituitary portal system. You can see in the diagram hypothalamo pituitary portal system. Through the neurons they are entered or through the portal system into the anterior lobe of the pituitary. You can see the arrow marks, they are following it. Second point is the hypothalamic releasing and hypothalamic inhibiting hormones are carried down the pituitary stalk by the hypothalamo pituitary portal system. You can see in the diagram the arrows are showing how these are carried down to the anterior lobe of the pituitary. Then here the hypothalamic releasing and inhibiting hormones increase or decrease respectively the release of the anterior pituitary hormones into the general circulation. This is the anterior pituitary part and this is the posterior pituitary part. So you can see in the diagram how the hormones are released at the hypothalamic neurons and then it is drained through the hypothalamo pituitary portal system into the anterior lobe of the pituitary, and here they have an effect on the release or inhibition of certain anterior pituitary hormones. Okay, so let's see what are the hypothalamic secretions. As we have said, they release certain releasing and inhibiting factors which are directly influencing the release of pituitary hormones. So let us see which are the factors the hypothalamus releases. So coming to the hypothalamic secretions, here you can see certain secretions. First of all is the GnRH, that is the gonadotropin releasing hormone. Second is the prolactin inhibiting factor, PIF. Third is the TRH, that is the thyrotropin releasing hormone. And fourth is the CRH, that is the corticotropin releasing hormone, and other secretions like GHRH, growth hormone releasing hormone, and the melanocyte releasing factors. So these are the secretions of hypothalamus. So we will study one by one. Here the main hormone related with the reproductive system is GnRH, that is the gonadotropin releasing hormone. So let's start about the GnRH first. Gonadotropin releasing hormone, GnRH. GnRH is also named as LHRH. LHRH is the luteinizing hormone releasing hormone. Okay, so GnRH is a decapeptide and is concerned with the release, synthesis and storage of both the gonadotropins FSH and LH from the anterior pituitary. You can see in the diagram, I'll show you the diagram. You can see here, this is a GnRH. Okay, chemical change of the GnRH. It is a decapeptide. It has the made up of 10 amino acids, and you can see the bonds between the 5, 6, 6, 7 and the 9, 8 are easily breakable. Hence the half-life of the GnRH is very short. It is 2 to 4 minutes only because of the cleavage of the amino acids bonds between the 5, 6, 6, 7 and the 9, 10 circulation. That's why its half-life is too short. It is acting only for 2 to 4 minutes. So here the GnRH is secreted by the arcuate nucleus of the hypothalamus in a pulsatile fashion. It has the effect on the release of gonadotropins FSH and LH from the anterior lobe of the pituitary. The divergent patterns of FSH and LH in response to a single GnRH are due to the modulating effect of the endocrine environment, especially the feedback effects of steroids on the anterior pituitary glands. This I'll explain you how the divergent pattern is there, like when FSH is rising, when LH is rising, but these FSH and LH are under the control of GnRH itself. Sometimes the feedback effect mechanism is also affecting the release of GnRH. So here like there are different types of feedback mechanism because of the ovarian steroidal hormones estrogen and progesterone are producing on the GnRH. So let us see about how these secretion of the GnRH is controlled. This we will see in our further slides. Here GnRH is secreted by the arcuate nucleus of the hypothalamus in a pulsatile fashion. This point is to be remembered. It is secreted in the pulsatile fashion. It is not secreted continuously, but it is secreted in a pulsatile fashion. And it is a decapeptide made up of 10 amino acids. You can here see In the diagram, the GnRH is released from the hypothalamus, which is acting on the anterior loop of the pituitary, and with this GnRH, it causes the secretion of the gonadotrophins, which are 2 in number, FSH and LH in the females. And you can see here the LH is acting on the ovaries. FSH and LH is acting on the ovaries to produce the ovarian steroidal hormones, estrogen and progesterone, hence causing the monthly cycles or the ovulation is there. This is how the HPO axis works together. GnRH is then secreted into the portal circulation in a pulsatile fashion. In a pulse fashion it is secreted. So what do you mean by pulsatile secretion? You may get a doubt. So what do you mean by pulsatile secretion? It is not secreted continuously or regularly. I said it is secreted in a pulsatile fashion. Yes. What do you mean the pulsatile secretion is? Pulse secretion varies in the frequency and amplitude. It varies in the frequency and amplitude at the different phases of menstrual cycle. That's why the pulsatile secretion is important. GnRH stimulates the anterior pituitary for the synthesis, storage and secretion of the gonadotrophin. Here the GnRH stimulates the pituitary to secrete, store and synthesize the gonadotrophins. There is decrease in the receptor sensitivity when the gonadotroph cells of the adenohypophysis or the anterior pituitary are exposed to the GnRH stimulation continuously, and this is called the down regulation. On the contrary, the intermittent exposure of the GnRH to the gonadotrophs increases the receptor sensitivity, and this is called as the upregulation. Are you understanding what I mean to say is, see what happens when the GnRH stimulation is continuously. If it is secreted continuously, then what happens? The receptors present in the anterior pituitary for the release. What happens when then they are exposed to the continuous stimulation of the GnRH? Then what happens? The sensitivity of these receptors decreases. Hence what happens? There will be down regulation. It will not act properly. It will not synthesize or it will not store or release the gonadotrophins, because the GnRH is continuously there, causing the decrease in the sensitivity of the receptors of the pituitary. So whereas the intermittent exposure of GnRH to the gonadotrophins, it increases in the receptor sensitivity, hence which is called as upregulation. But when it is released intermittently or in the pulsatile manner, then what happens? GnRH is released more. The gonadotrophins are released more, and the sensitivity of is also increases. This is called as the upregulation. This variable response of the anterior pituitary gonadotrophs to the GnRH has been utilized in the different clinical situations to get the therapeutic benefit. So this is called as a down regulation and up regulation. This is important, and we are using this concept in the therapeutical benefits, like we make certain GnRH analogs and we give these analogs. If we are giving continuously, then what happens? It will suppresses the release of FSH and LH if you give continuously. But whereas if you give in a pulsatile fashion, intermittent fashion, then what happens? It is increasing the release of gonadotrophin. So this concept we can use in the while giving the medications in the different clinical situations. So this is the down regulation and up regulation. Now coming to this part. Next are about the prolactin inhibiting factor. As I have told you, the hypothalamus releases different factors. One is the GnRH, this we have studied in brief about GnRH. Second is the prolactin inhibiting factor. The prolactin secretion from the anterior pituitary seems to be under the chronic inhibition of PIF. So in the girls, or unless and until the woman becomes lactating, this prolactin secretion is inhibited with the help of PIF, prolactin inhibiting factor. Okay, this is acting as a physiological inhibitor. It is also called as dopamine. So it is inhibiting the release of prolactin. Next is a thyrotropin releasing hormone. The hormone TRH is a tripeptide made up of 3 amino acids of the proteins. It stimulates the release of not only the thyrotropin but also the prolactin from the pituitary, whereas the prolactin inhibiting factor suppresses the release of prolactin, whereas the thyrotropin releasing factor releases the TRH, thyrotropin releasing hormone, which is acting on the pituitary to release the TSH, and which further produces the T3 and T4 from the thyroid, and it also releases the prolactin from the pituitary. Next is the corticotropin releasing hormone CRH. So this is a tetra decapeptide, and it is acting on the adrenal cortex to release the corticosteroids. And other secretions are GHRH, growth hormone releasing hormone, and melanocyte releasing factor. So this is about the secretions of the pituitary. Now coming to see the control of the GnRH secretion, how the neural hormonal control of the GnRH secretion is maintained. So it is modulated by the 4 ways, or it is controlled by the 4 ways, you can say. First one is the neurotransmitters and the neuromodulators. Second is the peptides. Third is the 3 types of feedback loop. One is the ultra short feedback loop, second is the short feedback loop, and third is the long feedback loop. So let's see one by one. Coming to the neurotransmitters and the neuromodulators, the control of the GnRH secretion is highly complex and dependent on a number of inhibitory or excitatory pathways involving the neurotransmitters and the neuromodulators with the interactions of the ovarian steroids. Okay, so here it acts as a neural input. So the important neurotransmitters, we'll see. The important neurotransmitters are catecholamines. There are 3 in numbers: dopamine, norepinephrine or noradrenaline, and the serotonin. These are the 3 main neurotransmitters. Norepinephrine exerts a stimulatory effect on the release of the GnRH, while the dopamine exerts its inhibitory effect. Serotonin plays an inhibitory role in the GnRH. So among these 3 main catecholamines or the neurotransmitters, only the norepinephrine or the noradrenaline has a stimulatory effect on the release of GnRH, whereas the dopamine and serotonin exerts a inhibitory effect on the release of GnRH. Okay, so these catecholamines are capable of changing the frequency of the GnRH pulse. Thus, the pharmacological agents, psychological factors which affect the brain catecholamines are likely to affect the pulsatile release of the GnRH. Okay, so this is about the neurotransmitters. Neuromodulators, neurotransmitters transmits the impulse from the one neuron to the other, hence the name neurotransmitter is mentioned. Neuromodulators, they modulate the regions or the circuits of the brain. They affect a number of neurons, causing a modulation of that group. So neuromodulators alter the neural activity by amplifying or dampening the synaptic activities. So this is the difference between the neurotransmitters and the neuromodulators. Neurotransmitters are transmitting the impulses from one neuron to another, whereas the neuromodulators, they just modulate the regions or the circuits of the brain. So coming to the neuromodulators, they are the endogenous opioids, peptides, and the prostaglandins. So here 3 main neuromodulators are there: endogenous opioids, peptides, and the prostaglandins. Coming to the endogenous opioids, they are the mainly important endogenous opioids are the endorphins, encephalins, and the dynorphins. So these are the 3 main opioids: endorphins, encephalins, and the dynorphins. So they collectively, the opioids, they inhibit the release of GnRH, thereby inhibiting the gonadotropin secretion. So inhibit, so negative mechanism, negative feedback effect will be seen here. They inhibit the release of GnRH, the endogenous opioids, which are 3 in number: endorphins, encephalins, and the dynorphins. The endorphins may suppress both the dopamine and GnRH pathway, leading to increase in the prolactin secretion. Increased endogenous opioids have been actively implicated in the hypothalamic amenorrhea and the elevated prolactin levels observed in association with the stress and exercise. Prostaglandin E produced locally increases the release of GnRH. Coming to the prostaglandins, the endogenous opioids are over. They have the negative effect, whereas the prostaglandin E produces locally increases the release of GnRH. So prostaglandins increases the release of GnRH. Now coming to the peptides. Peptides, a number of peptides like activin, inhibin and follistatin. Follistatin, these are the three main peptides, activin, inhibin and the follistatin. These are produced by the GnRH from the anterior cells as well as from the granulosa cells of the pituitary. Activin stimulates, but the follistatin inhibits the GnRH secretion. Inhibin blocks the GnRH secretion. So here only the activin is stimulating the GnRH secretions, whereas the follistatin and the inhibin blocks the GnRH receptors. Okay. So this is the diagram showing the neurotransmitters and the neuromodulators in the control of GnRH secretions. You can see here in the first point the neurotransmitters, which are 3 in number: dopamine, norepinephrine and the serotonin. As I have already mentioned you, only the norepinephrine or the non-adrenaline has a positive effect on the release of GnRH, whereas the serotonin and dopamine, it inhibits the release of GnRH. Coming to the neuromodulators, which are 3 types, mainly the endogenous opioids, neuropeptides and the prostaglandins. Here the opioids has the negative effect. Endorphins, encephalins and the dynorphins have a negative effect on the release of GnRH, whereas the neuropeptide and the prostaglandins have a positive effect on the release of GnRH. And the peptides also have a peptides. Among the peptides there are 3 types, mainly the activin, follistatin and the inhibin. Only the activin has a positive effect, whereas the follistatin and inhibin has a negative effect on the release of the GnRH secretion. Now coming to the other methods of the control of the GnRH, there are 3 feedback loops: ultra short feedback loop, short feedback loop and the long feedback loop. So coming to the first, ultra short feedback loop. Why it is called as ultra short? Because here it is limited to the hypothalamus itself. How it is? Let us see. It refers to the auto-regulation of the releasing hormone of the hypothalamus on its own synthesis. If the GnRH production is more, it will suppress. If it is less, it will stimulate its further production. GnRH can regulate the concentration of the pituitary receptors. You can see in the diagram, it is the ultra short. Ultra short means it is present within the hypothalamus itself. So hypothalamus itself regulates, means it is the auto regulation of its own synthesis. So whenever the GnRH secretion is more, it will suppress automatically. That is the auto regulation. Whenever it is less, it stimulates more. This is called as the ultra short feedback loop, which is present within the hypothalamus itself. That's why the ultra short. It is not involving the other organs or the other tissues or the hence it is called as the ultra short feedback loop. Next is the short feedback loop. Short feedback loop, here it is involving the pituitary. Okay, so it is related to the secretion of GnRH by the interplay between the pituitary and the hypothalamus, and here the neurotransmitters are also involved. So here what happens, the FSH and LH believe to exert a negative feedback effect on the hypothalamic production of the GnRH. So here what happened, the pituitary releases the FSH and LH. So whenever the FSH and LH are more in the circulation, automatically it exerts a negative feedback effect on the hypothalamus. Thus it is called as the ultra short, because it is related with the pituitary, hypothalamus to pituitary or pituitary and hypothalamus. So whenever the pituitary secretions of the gonadotrophins FSH and LH are more, it exerts a negative effect on the release of GnRH. Whenever the FSH and LH secretions are less, it exerts a positive effect. So in this way it is called as a short, because you can see here it is related with the hypothalamus and the pituitary. So this is called as a short feedback loop. Next is about the long feedback loop. It is related with up to the ovaries. Here you can see in the long feedback loop, you can see the hypothalamus and ovaries are related. Ovaries secretes the sex steroidal hormones like estrogen and progesterone. So here what happens, the secretion of hypothalamus, GnRH and pituitary both are influenced by the sex steroids or the estrogen and the progesterone, which are secreted from the ovaries. So sex steroids estrogen and progesterone, they exert a negative effect throughout except in the pre-ovulatory period, where a high and sustained level of estradiol or the estrogen exerts a stimulatory effect in the release of GnRH through the catechol-estrogen. So what does it mean? So whenever there will be more or increase in the number of estrogen and progesterone, it is inhibiting both the hypothalamus as well as the pituitary. Except in the pre-ovulatory period, before the ovulation, whenever the estrogen level will be high, it causes a stimulatory effect, so that there will be release of LH surge is needed for the ovulation. So only in that case, pre-ovulatory estradiol level will be more. It causes a positive effect to release the LH. So LH surge is needed for the ovulation purpose. So except in that case, in throughout the cycle, it is causing the inhibitory effect. So whenever the estrogen levels are more, progesterone levels are more, the pituitary and the hypothalamus is suppressed. Except in cases of the pre-ovulatory period, where a high and a sustained level of estradiol exerts a stimulatory effect in the release of GnRH. So this is about the long feedback loop. You can see here how the long feedback loop, short feedback loop and the ultra short feedback loop is acting. A is denoting the ultra short feedback loop. It is limited up to the hypothalamus. B is ultra short is C. A is the long feedback loop. You can see here the hypothalamus. Target organs are nothing but the ovaries. And B is the short feedback loop. It is related with the hypothalamus and the pituitary. C is the ultra short, which it is only limited up to the hypothalamus itself. This is the auto regulation of the hormones. In the diagram also you can see here the long feedback loop, short feedback loop and the ultra short feedback loop. Yeah, remaining 2 points are there. Here the coming to the nature of secretion of the GnRH. So GnRH release and secretions are tonic and cyclic and pulsatile. There are 3 types of secretions: tonic center, cyclic center and the pulsatile release. So the tonic center is primarily controlled by the negative feedback effect of the sex steroids. So here tonic center is through the tonic center, this is related with the long feedback loop. So long feedback loop is controlled by the feedback effect of the sex steroid. So here the tonic center is acting, the long feedback loop is acting on the tonic center, whereas the cyclic center is stimulated by the positive feedback effect of the pre-ovulatory sustained and high level of estradiol, resulting in the increased GnRH secretion, whereas the pulsatile release is the secretion of GnRH into the portal circulation is presumed by regulatedly by the neurotransmitters and the neuromodulators. So what do you mean by pulse secretion? Pulse secretion of the GnRH or the pulsatile secretion has amplitude and a frequency. What do you mean by amplitude? The amplitude means the amount of the GnRH which is delivered into the portal circulation. Amount is the amplitude, whereas the frequency means at what interval the GnRH is delivered into the portal system. So it is the interval. How many times it is delivered is the frequency. How much is delivered is the amplitude. So the frequency is rapid. This is the important point. The frequency is rapid in the follicular phase, about 1 pulse per hour, and lower in the luteal phase, about 1 pulse every 2 to 3 hours. In the follicular phase, the amplitude is low, but it is high in the luteal phase. So frequency is more in the follicular phase. Follicular phase, the frequency of GnRH is more. It is 1 pulse per hour. Every 1 hour, you will get the 1 GnRH pulse in the follicular phase. Whereas in the luteal phase, it is 1 pulse in the 2 to 3 hours. In the follicular phase, the amplitude is low, but it is high in the luteal phase. I'll show you the presentation of it, then you will come to know. Here you can see in the diagram, this is a graphic presentation, you can easily understand. You can see in the follicular phase the GnRH pulsatility or the frequency is more, whereas its amplitude is less. Whereas in the luteal phase, its frequency is less, whereas its amplitude is more. And you can see here how the LH is rising and sustained release of the FSH is seen here. Rising in the just before the ovulation, the LH surge will be there. You can see high frequency in the follicular phase, whereas a high amplitude in the luteal phase, and it is secreted by the arcuate nucleus. Yes, next point is the GnRH action on the anterior pituitary. It is called as the self priming of the gonadotrophins. Here what happens, the GnRH is not only involved in the release of the stored FSH and LH, but it is also important in the maintaining the synthesis of the gonadotrophins. So what happens, GnRH on reaching the anterior lobe of the pituitary, it stimulates the synthesis and release of both the LH and FSH from the same cells or gonadotrophins in the pituitary gland. So here the lower GnRH pulse frequency, it favors the FSH secretion, and the higher frequency favors the LH secretion. The response has been termed as a self-priming. It has got a cyclic relationship, and it is greater with the higher levels of estrogen. Estrogen preferentially induces more LH than the FSH release. So here the GnRH is not only helping in the releasing, but it is also helping in the maintaining of the gonadotroph FSH and LH. Coming to the physiological control mechanism of gonadotrophin release, the two poles of gonadotrophins are present in the pituitary. One is the primary pole and another is the secondary pole or the reserve pole. So one is readily releasable by the initial exposure of GnRH. So first of all, whenever the GnRH is released, the primary pole which contains the gonadotrophins, from that it is released, and the second one is called as the reserve pole or the secondary pole. So GnRH makes this large reserve pole to be readily released by the subsequent repeated exposure, and this is a suggestive of a transfer of gonadotrophins from one pole to the another. So first of all, with the initial exposure, the gonadotrophins are released from the primary pole, and with the subsequent exposure of the GnRH, this reserve pole or the secondary pole is activated. And the last point in this is the mechanism of action of GnRH on the pituitary receptors. So GnRH binds to the specific receptors on the cell membrane of the gonadotrophs. Gonadotrophs are the cells of the pituitary which releases anterior pituitary which releases the FSH and LH. So within a second there is activation of the enzyme adenocyclase, which catalyzes the conversion of ATP to cyclic AMP, and this AMP receptor protein complex then activates the protein kinase C, and here the protein kinase C along with the calcium, it causes the phosphorylation and activation of specific enzymes. So here Ca, calcium, protein kinase C and CAMP, they interact together to release the stored FSH and LH and their subsequent biosynthesis. However, if the GnRH continues to be infused, the gonadotrophin secretion is inhibited, probably because the receptors are saturated and are unable to stimulate the release of second messenger. This is known as the desensitization or the down regulation. You can see in the diagram, first of all the GnRH, whenever the GnRH is released, it interacts with the GnRH receptors present in the cell membrane of the gonadotroph cells or the anterior pituitary cells. Then adenyl cyclic complex will be created, then it releases ATP, and then it is becoming the CAMP, and then what happens, there will be a release of protein kinase C. Then protein kinase C, it does the phosphorylation, that is, it is converting the proteins into the byproducts. Okay, and in this phosphorylation, protein kinase C along with the calcium and the complex CAMP complex is acting together to release this gonadotrophin. So this completes the hypothalamus and it's released in the HPO axis. So we will be continuing about the pituitary in our next classes, pituitary and the ovaries, how they are, how different they produce as different hormones, and how they are acting together in the HPO axis. Thank you.